And we already talked about this, thank you. We already talked about the vision, and I'm going to go a little bit more in detail, but what you see in reality is like this little car over on the left. You see the car out there, but in reality, it's the signal that comes in your brain that you have to interpret. So the image that's coming to your brain, this is what your eyeball is seeing. Your eyeball only has a certain number of quote-unquote pixels it can detect. So you see this kind of blocky, not perfect image, and your mind cleans it up and purifies it, makes it look like the car that you are familiar with. Like I said, a lot of these things, in your mind you're going to think, no way, but I'm hoping throughout, like yesterday and today, I'm proving to you that the things that you think are real are actually just created by your mind. And I'm not getting all matrixy and stuff like that on you, but it's just interesting to understand how your mind thinks. In undergrad, I did psychology, and it was just amazing. All the things that they said, this is the average person, this is the way they behave, and the stupid things the average person does. And you know what? We all say, no, we don't act like that. But when you look at the statistics and who does what, you're amazed because usually you're in the average. I mean, it's just crazy looking at all the things I've done, and I think, I'm unique because of this, but I'm not. I'm just average. Your brain's the same way. You think, no way my brain's different than with the way I think of it, but you know what? You are. You're just kind of an average person. No offense. All right, so when you look at the structures, this is anatomy, and remember, anatomy, am I going to test about anatomy on exams? No. This is just to get you back and up to speed. So when you look at the protective mechanisms, you got the eyelids to basically brush things across the eye to distribute the tears. The tears are there with some special stuff called lysozyme. You know what lysozyme is? Do you remember it? Do you remember what a lysosome is? Mm-hmm. It's the little organelle inside the cell that does what?
Oxidize.
It breaks things apart, right? It's like the stomach. It kills things. When a cell eats, like a macrophage eats bacteria, it eats the bacteria. The lysozyme digests the bacteria. You have that chemical in your tears. Why?
So you don't get eye infections?
Yep, to try and kill bacteria. So it's there to help try and kill bacteria. Of course, if you get overexposure to bacteria or you're decreasing your tears and things like that, you're more susceptible to infections like pink eye. But yeah, it's there to help kill the bacteria. And then you have eyelashes to catch fine debris as it's coming towards you. Not like a baseball, but like dust particles to try and catch. When you look at the eye itself, this you do have to know. There are three major layers of the eye. There's the outside, the white part of the eye. What do we call that? The sclera. It's white for a reason. It's there to deflect all of the light it can. It's trying to get rid of all of the light so it doesn't go into the eyeball, except right at the tip. And what do we call the sclera when it comes to the front of the eye? It starts with a C. The cornea. Yep. So as it gets close to the front of the eye, it actually becomes clear. So it's all white around here until up here it actually becomes clear. There's no break. It's continuous tissue. It just changes its color, so it's transparent. And then just underneath that, you have a layer called the choroid layer. And the choroid's really important because it gives you blood vessels all the way around the eye. It's full of capillaries and little arteries and veins. It distributes all the nutrients. Because even your cornea, it's alive. It's changing its structure. It's constantly needing to be fed and provided oxygen and everything to keep it alive. So the choroid brings all the blood up here and nurtures the layers of the eye. And then the inner layer is the retina. And the retina, of course, we're familiar with rods and cones. They're the little detectors, the receptors for light. So those are the three major layers. When you look at that choroid, by the way, just like the sclera, when it gets to the front, it doesn't just stop. It actually becomes these ciliary ligaments and then the lens. And then the lens, of course, is there to help bend the light, so change shape of light or refract light. The retina will come all the way up here, and the retina stops right about here. But the retina, you have lots of rods all along the outside, tons and tons of rods. And then as you get closer to the middle, you get more cones. And the cones are there for detecting what? Light and dark or colors? Colors. The way to remember that, I always think of snow cones are multicolored, right? So snow cones in the eye are multicolored. The rods are there for light and dark. Presence of light or absence of light. They detect black and white. When you look at the cones, they're there to detect three colors. So everything you see as far as color is only three colors. Red, green, and blue. And we'll talk about that in a few slides. So there are the major layers. The sclera, the choroid, and the retina. And you need to remember the structures on them. The fluid that's inside, you've got the vitreous back here and the aqueous up here. Those just circulate nutrients. And the vitreous humor actually gives shape to the eyeball. It's a little bit thicker. When you're in anatomy, you slice the cow eyeball apart. You probably remember that. It's kind of like, almost like liquidy jello. So it helps give shape to the eyeball. Anybody ever seen floaters?
Yeah.
So they can be normal. If they're excessive, then it's a problem. But floaters, you can actually have little proteins that are floating in here. And it's weird because sometimes when you you're like, it sounds like this, and you look and you see the little floaters coming through. They're proteins. They freak you out. I'm always worried because you can actually get parasites in your vitreous humor. And I'm always thinking, oh my god, I hope that's not a little worm.
Or like, it's weird because it'll like slowly move, and then if you look again,
Yep,
it'll,
and
like,
it shifts.
Yeah.
Right. Yeah, they're crazy. The eye is an amazing structure. All right, so here we have the structures on the front side. Remember, what is this? It starts with a C. Cornea. There's the lens. What's this thing that's colored? Iris. What's the center part of the iris? The pupil. And the iris is just like a diaphragm on your camera that allows light to be adjusted. So it can open up and let more light in or squeeze down and let less light in. And then here you have, when you're nurturing this stuff, remember this fluid is providing nutrients, it's bathing the lens and the cornea. So the fluid's made up here, it gets shifted out, circulates around, it's called aqueous humor, by the way, and it comes back down and leaves this thing called the canal of Schlemm. The reason I'm pointing this out is because there's a disease out there they usually check for when you get your eyes checked, that if this canal of Schlemm is blocked, or it's not working properly, or clogged, or whatever term you want to use, this fluid just keeps getting made. What's happening to the pressure in the eyeball? It's getting bigger. Remember this choroid layer? What's in the choroid? Blood vessels. The more pressure you have in here, what's going to happen to these little tiny capillaries and arteries and veins? They get smashed. Yeah, they get smashed, so they can't circulate very well. What's going to happen to all this tissue when you're not getting good circulation? It's going to start dying. Yeah, it's going to atrophy and then die. That disease is called, anybody know what that disease is called? Glaucoma. Yeah. So glaucoma slowly compresses, the pressure in here compresses the blood vessels, and then they start dying off, and then vision's lost. Yeah, I'm not even going to make any comments about smoking weed. We'll just leave it out there for you guys to think about. All right, so the function of the choroid, if I asked you a question like this, what's the function of the choroid? Remember, this isn't the structure of the choroid, this is actually the function. So what layer has receptors for light?
Sclera, isn't it?
Cornea.
Oh.
Which layer has receptors that detect light?
Retina.
The retina. Yep, so the retina, how about number 2? Which layer is the protective outer layer? The sclera. Yep. How about which one gives the shape of the eyeball? That's the vitreous humor, right? The vitreous humor. And then how about provide blood supply to the retina and sclera? That one's the choroid. Yeah, so it's the choroid. All right, the iris. The iris is so cool, and I wish I had time to go into lots of detail about it, but what we take for granted is layers. Like our skin, we don't even always think about how many layers the skin is. The iris is the same way. The iris actually has 5 layers to it. It looks like one little colored layer, but there are 5. If you looked really in depth, you could see, it has like 3 dimensions to it. But there are 3 really important layers. The front colored layer has pigments in it that give you your eye color. But there's really only one pigment, and it's this greenish brown pigment. Either you have it, you have lots of it, or you have none or very little of it. It's all the same pigment. So people that have lots of this greenish brown pigment, guess what color their eyes are?
Greenish brown.
Brown, like a real strong brown. If they have less of it, what color does it start turning? More of a greenish color. And it all depends on the amount of pigment. The way it bends the light or bounces light makes their eyes look more of these shades in between. If they have none of that pigment, the light goes right through that layer and it bounces off the next layer. And the next layer actually has these pigments that are like a purple color. They look like this. So they're actually a purpley color. When light bounces off of it and comes back, it actually changes it to what color? Blue. So if they don't have that pigment up here in the first layer, so if there's no pigment in that first layer, the light bounces off the purple, comes back, and they have blue eyes. So everybody actually has blue eyes, but you have a brown layer in front of it that blocks that light.
Is that why all babies start out with blue eyes?
A lot of babies start out with blue eyes, yeah. And then
99.9%.
And then the last layer is actually blood vessels, because this is still alive, right? So all the way in the back, you have to have blood going in. So if you are born without the ability to make the pigment in the front or the blue pigment in the next layer, what are you actually seeing for color? Blood, which is like a reddish pink. Yeah, not blue and not brown. So you see, and what do we call those people? Albino, right? So they don't have the pigment in their iris, so the light goes right through, and you're actually seeing light bouncing off blood vessels coming back. Like rabbits are the best example because they're very brilliant pink. So it's kind of cool how many layers you have. The second thing about the iris is it actually has 2 layers of muscle. It has a muscle that's circular that goes all the way around. It's a circular, smooth muscle. Do you have control over it? It's smooth. Nope, involuntary. It's a muscle, so when it turns on, what's it do? Does it expand or does it contract? Muscles only contract, right? When they relax, they flop open. But so when I squeeze this muscle, it squeezes in. Would that be associated with your fight or flight or your relaxation?
Fight or flight.
When your pupils get really small and they constrict.
Relax.
Relax, right? When you're afraid for your life, what happens to your pupils? Huge, right? The other ones are radial. They radiate out like this. So when you're afraid for your life, they pull the iris out. Why are they pulling the iris out? So that your pupil does that. To let more light in, so you can see more of your surroundings and better. You can see in depth and everything when you're freaking out. You ever seen like black, what do they call it? Like night vision, that's what it is. Have you seen those night vision pictures of people sitting in a dark room and their pupils are just huge, they look like vampires? It always freaks me out. What's kind of cool is these reflexes, the fight or flight and the relaxation reflex, can be stimulated by your skin. What's kind of neat, and if you want to do this, you can look at your neighbor, and if you run your finger lightly at the back of their neck, the nape of their neck, while you're watching their pupils, watch for something to happen. In fact, I could actually feel my eye changing when I did that. What you're doing is you ever had a feeling like it makes the hair on the back of your neck stand up? Is that a fight or flight or a relaxation instinct?
Fight or flight.
It's fight or flight. So what should happen to their pupils when you're looking at them?
They get bigger.
They get bigger, yeah. Their pupils should actually open up a little bit and then come back to normal size. You're triggering that fight or flight instinct by doing something like that. It's kind of cool how your body can actually manipulate your brain. You can do, and if that didn't do it, just pinch the back of their neck really hard. But that'll do it too. Not enough to draw blood, but just enough to give them a little ow. Okay, oops, I swear I changed this. It's not. All right, so the lens. There are two things that bend light. One's the lens, and the other one is the cornea. What's not normally understood is that the cornea actually does 2/3 of the light bending. But what do we give credit for bending light to? The lens. The reason we give so much credit to the lens is because the lens is easy to change its shape. It's very flexible. You can stretch it, make it flat. You can squeeze it, make it more round. But the cornea doesn't want to change very much. In fact, people that have problems where they can't bend the lens very well, what they do to change the cornea is what?
Squint.
Squint. Yeah, they're squeezing their eyeball down. They're actually forcing their cornea to take a new shape. But the cornea does most of the refraction of the light. Refract means to bend light. Reflect means balance it back, if you didn't know the difference. But this refraction is bending the light and focusing it back on the retina, onto the rods and cones specifically. So 2/3 of that bending comes from the cornea, 1/3 comes from the lens. And what you're trying to do is you're trying to get that light to focus perfectly on the retina. So when you see an image, points should come right back and focus right on a couple rods and cones so that your brain can detect that specific line. What was the name of that process where you can detect a specific point? Was it receptive field or was it lateral inhibition? Lateral inhibition. So when a piece of light hits this receptor, it will shut off its neighbors so that your brain can actually make a black and white outline, or actually a colored outline of whatever the image is. But the problem is if the lens isn't bending the light properly and that light starts spreading out and it affects a bunch of points, your vision becomes blurry. Same thing here. If it focuses too early, it makes a blurry image on the retina. If it focuses too late, it makes the same blurry image. I'll show you how that works. I'll show you the diseases actually. The process of changing the lens is called accommodation, Star. So accommodation is changing the lens. And the steps are written here, but I'm going to show you the picture. So here you have that little ciliary body that I was talking about. It's a group of smooth muscles. Do you have voluntary control over it? No. Nope. It's involuntary movement. When you pull on this muscle, it pulls these little ligaments, and the ligaments hold onto the lens. And what's it going to do to the lens if I pull on it? Stretch it out. It makes it flat. So if this is a normal lens and I stretch it, boink, it flattens it out. It's like bending a magnifying glass. It's like flattening it out. The better magnifying glasses that magnify a lot better are what? More rounded or very flat, like a piece of glass?
Rounded and thick.
More rounded. If you look at a flat piece of glass, we call them windows, right? Do they really bend the light and make things look weird? No, they're perfect. But if you had a rounded window, one that came in towards you and then came out on the other side, everything would be magnified and maybe even flipped upside down. So when you're stretching this out and you're making it more flat, you're actually making that image a little bit different. So if I want to see something far up, if I want to see something that's far away, I want to see it a little bit better, I'll actually flatten that lens and it makes everything in the background clear. How many people here own cameras and enjoy photography? It's kind of cool. It's the same concept. The camera mimics your eye. So if you have lenses that are flatter, you actually get more of a wide image, a more clear image. The more round that lens is, the more close up you have to look at things. So if I relax this muscle, it makes, by elastic properties, it makes that lens more round. What am I going to look at better when the lens is round?
Things close up.
Things that are close up. When a baby's born, their lens is more round. What do they see better? Close up. And as we age, what happens to the elasticity of our lens? It gets worse. So it gets more flat. It doesn't bounce back to that round shape. So as we age, what kind of vision do we see better? Close up or far away? Far away. My grandfather always denied that his vision was getting worse. He said, because he'd read the paper like this. He said, my vision's not getting worse, my arms are just getting too short. So he'd be reading back like this. So we get farsighted as we age. So when you look at the pathway, now that you know the different layers, you understand how accommodation works because of the ciliary bodies and the lens. If I ask you a question like this, light waves pass through all of the following structures except what? What's the only thing that it doesn't pass through? Right. What's the whole point of the sclera? To prevent light from moving through. What color is the sclera? It's the white of your eye. It's there to block light from getting through. So that all the light going into the retina is actually coming right through the pupil. So it goes through the cornea, then the aqueous humor, and then the lens, and then what other humor? The vitreous humor, and then it hits what layer of the eye? The retina. Okay, so here are a couple of disorders. Disorders can happen for a couple reasons. Number one is it can happen because the structures that refract the light are malformed or incorrectly formed. What are those two structures? The two structures that bend light for you. The lens and the cornea. Yep. So let's go with the lens first. If the lens is too flat, what kind of vision do you have? If it's a flat. Farsighted. So you see things far away better. So when you're reading things, you'll have a tendency to go way out like this. If the lens is too round, what vision do you have? Nearsighted. So babies start with a round lens, and as we age, they get flatter and flatter. And if it helps you remember, just remember that round, flexy lens, babies, they have to see things really close. But as we age, they get further and further away because the lens keeps getting flatter and flatter and flatter. Right. The other thing that can be changed is the shape of the eyeball. So the lens can be dysfunctional, the shape of the eyeball can be dysfunctional. So if you have a normal eyeball, looks kind of like this, if your eye is too long, you actually get nearsightedness. Because it's trying to focus, look at the distance, it's focusing the light at this point. When your eyeball is too long, now it's still focusing at about the same point, but it gets blurry the further back. If you have
astigmatism?
This is actually just nearsighted so far. And I think in your notes it says myopia. Is that written in there somewhere? M-Y-O-P-I-A. If it's not, go ahead and jot it down. It's myopia. That's nearsightedness. And you can see from this picture how the ball is actually more clear than the kids. And the fence is really blurry. It's nearsightedness. They see things closer up, a little bit more clear. And if you play with cameras and you understand that, they call this bokeh in the background, where it's all blurry and fuzzy. It makes the pictures look cooler, where you focus on something close and everything in the background looks really fuzzy. And then farsighted is just the opposite. I mean, you can hold this paper up close to you, but everything seems blurry. As you slide it backwards, it becomes more clear. And they call that hyperopia. So hyper, you can think of as above or further or greater than or whatever. It means that their vision's better when the distance is greater. And then like you just said, there's another one called astigmatism. And astigmatism primarily affects the cornea.
It's like pokes it out, right?
It actually makes it all kinds of different shapes. But yeah, it can bulge it out. It can do just one area. So if you're looking at a round lens, it may have just a flaw over here. It might have one just down here, one over here. It might have them all over the place. So it can be all over. When the light hits those areas, it bends it in really strange ways. And later we'll do a lab. We'll have a makeup lab that's didn't fit anywhere in this section. But we'll look at vision. We'll talk about astigmatism and we'll test your vision to see how it works. But if you look at certain charts, like if you look at a clock, certain numbers on that clock might seem a little bit weird compared to the other ones because of the way the light's bending in that area of the cornea. It's called astigmatism. Can you get that corrected?
No, not easily.
With a laser, yeah, they burn your eyeball. Nothing like the smell of flesh burning, in the doctor's office. But yeah, they can correct most of them with the laser. It used to be they couldn't make contact lenses because, think about it, a contact lens is round. It doesn't have an up and a down. So when they put it in the eye, if you're trying to correct this area up here, what if the lens came in the wrong way? Now you've screwed up this area and this area. So what do they do, the new contact lenses, to make up and down? Put a little weight on the bottom. Yeah, there's a little weight on the bottom, so gravity lines it up for them. I love science. It's so cool when you step back and think about how things work. So there are some disorders. Myopia is what? Far or near? Near. Hyper would be far. And then what's the one that the cornea is defective? Bent and astigmatism. Yeah. Okay, so the layers that you're passing through. So now we've talked about light coming into the eyeball. Now we're going to hit the retina. This is a really close-up slice of the retina. So here you have the retina in the back. If we look, this is amazing to me. So if you take pictures, how do pictures look if you have a bunch of debris on the front of the lens, like dust and particles built up? They look blurry and crappy, right? This is amazing. These are the detectors. The rods and the cones are all the way back here. This is the back of your eyeball. For light to hit those, it has to go through all of this nerve tissue and then hit the rods and cones. So light penetrates through here, comes moving back, bounce, 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 bounce. Finally, it hits this. So all this crap sitting on the front of your receptors should give you a blurry image. But your brain takes that blurry, fuzzy, junky image and actually modifies it, cleans it up, and gives you a perfect, crisp image. So the layers you want to get familiar with, you have the front ones that are called the ganglion cells. Okay. You actually have another one called amacrins, and then you have these bipolars, and then you have the horizontal, and then you have the receptors. The three main areas are these three, the yellow, the blue, and then the rods, cones. So the rods and the cones are the actual receptors. The light has to hit these. If you don't have these, you don't have that vision. People with red-green color blindness have flaws in the red and the greens. They're missing pigments, or they're missing the ability to make that specific one. So they see things other than that color light. These are the connectors. So remember how I was talking about lateral inhibition? When this rod gets fired, what's it going to do to these two neighbor rods? Shut them off. When it gets fired, this is the cell that's responsible for shutting the neighbors off. So it's going to turn off the neighbors so that you get a specific pinpoint. And then this last set of cells are called the ganglions. And these are so important because the ganglions sit on the inside. They all come together. They're all moving the same pathway, and they actually merge to the optic nerve. They form the optic nerve. So all of them come back here to this one point and leave the cell, or leave the eyeball. So when you see an image, you have about 120 million rods and cones, sorry. You weed them down into 1 million ganglion cells. So like 120 of these will all go to the same ganglion cell. Remember yesterday when I was talking about having 4 receptors, when we talked about receptive field, like 4 receptors that all go to the same signal outside of the room? If I hit any of those 4 receptors, can your brain distinguish between the 4? No. That's what's happening with 120 receptors all going to one ganglion cell. That's another reason your vision's so blocky, because when light hits anywhere in this area, your brain can't distinguish which specific area it is. These guys will shut their neighbors off and say, hey, we're in this area somewhere. So it all comes down to these ganglion cells. If you had 120 million ganglion cells, one for every receptor, the back of your eyeball, your optic nerve, would be exactly the same size as the back of the eyeball going into your brain. So it's a way for your body to actually save some space. It's kind of a fascinating, cool way that this works. But the rods, the cones are the receptors, the bipolars are the ones that help split and turn things off, and then the ganglion cells are the ones that form what structure? The optic nerve, yeah. So they actually carry the signal to the brain. Right, special areas, I already talked about the optic nerve. What's also incredible about the optic nerve is that where the optic nerve goes out the back of the eyeball, there are 0 rods and 0 cones. So how's your vision right there? There are no receptors. There is no vision there. It's a blind spot. It's technically a blind spot. Did I have you cover it up? The plus and the circle on your paper, your white paper. What's incredible about your brain is, remember, you perceive things, you plug in pieces. There is a black nothing hole right in your vision. Wherever the light hits on this, that's just a dead area. You can't see anything. But what your brain does is it detects all this signal, and then plugs in what should be there. So with that, the little plus and minus, or sorry, plus and circle here, if you look at the plus sign with your right eye, focus on the plus sign, and then move your head closer. Keep focusing on the plus sign. As you move it closer to the paper, that black dot should disappear. if you keep twitching your eye over to see the black dot, it'll never disappear because your brain knows it's there. But if you focus specifically at the plus sign and you move your head closer to the paper, keeping your eyes straight on that plus sign, by the way, you have to cover your left eye. As you keep your eye on the plus sign, keep moving closer, suddenly that black dot will just disappear. It just turns white there. Because your brain's plugging in all the information saying, well, it looks like paper, I can't see anything there, so everything around it must be paper. And then you can do the same thing with the other eye. This is a monocular test. You cover your right eye, focus on the circle with your left eye, and then move in. You'll have the same thing happen. The plus sign will disappear. So always keep, the one you want to disappear, always keep to the outside part of your body. And that's because you're lining up that image that disappeared, you're lining it perfectly into the optic nerve. Just the opposite of the optic nerve is, did you have a question?
All this 3D stuff that's coming out, is that more like perception that we're
Actually, it is. Your brain knows that you have two eyeballs, and each of the eyeballs are slightly off, so it takes a signal from one side and a signal from another side, and it kind of like interprets it. And I actually have a couple slides we'll test on this in a little bit. So yeah, 3Ds don't work very well if you only have one eyeball. Just makes it a little more challenging. All right, so the fovea is exactly the opposite of the optic nerve. The fovea is where you get the most intense perception. It's the best acuity is another way to say that. It has the most dense area of cones. What do you see best in here? Black and white or color?
Color.
Color. This is incredible, but it has a low concentration of rods. So this is interesting too. Sharpshooters, when they're taught to shoot at night, when you stare at something, the light comes in during the day, and all that color light focuses right there on the fovea, and you get this really good, clear picture. But at night, do you see things in color very well? No. So what they teach sharpshooters is at night, you look at what your target is, and then you shift your eyes slightly off to the side. Why are they shifting their eyes slightly off to the side?
So it doesn't go into your blind spot.
Exactly. So they don't want to hit the blind spot, they don't want to hit the fovea. At night, all they're seeing is black and white. Out here is where you have a high density of the rods. All out here, slightly peripheral to the fovea, is where you get better night vision. So you shift off to the side and you can see things a little better. Birds actually have two foveas. They have one on the top and one on the back. Why do they have one on the top? Think about it a second. Why does a bird have a fovea, a very clear color vision at the very top of their eye?
So it can see where it's flying.
So where it's flying, you're right, on the back. But why up there?
It's predator, so it can see down.
So that they can see what's below them just as clearly as what's in front of them. So as they're flying over, they don't have to keep looking down to see for worms, right? Because if they look down for a worm, what's going to happen?
They'll fly down.
Right? They might slam into a tree. So they can keep flying straight forward, but they also see down. It's kind of a really cool process if you ever get a chance. Owls have really good vision. What's interesting about owls is they can't move their eyeball. They have no muscles to turn their eyeball to the side. So what do they have to do?
Look.
That's why an owl can almost turn its head 180 degrees. Because it can't turn its eyeballs. They're fixed straight ahead. So it's just constantly doing this. All right, so if I asked you what part of the retina would be best acuity, for the best acuity? Yeah, the fovea. So remember, the ciliary body helps you with what process? It stretches the lens. What's the process of stretching the lens to see better? Accommodation. Yep. The blind spot is where what enters the eye, or sorry, what leaves the eye, optic nerve. Fovea, like you said, this is the one that's best acuity, and the iris does what for vision? Adjusts the amount of light coming in. All right, and then the pathway. So we're going to do some visual tricks in just a couple slides. But another thing that's cool about the eye, each eye actually talks to both sides of the brain. But how it talks is interesting. Everything in your left visual field comes over here, hits the lens, and is refractor bounced over to the opposite side of the eyeball. So everything in your left visual field will hit the right side of the eye. It doesn't matter which eye. Left visual field will hit the right side of that eye too. But what your brain does is right here. What's this crisscross called? It's the Greek letter X, which is called chi. It's the chiasm. So this crossing over, this is a processing point. It takes the right side of this eye and the right side of this eye and directs them in the right direction, the correct direction. So your left visual field comes into this side of the right eyeball, comes all the way down to the chiasm, and then it crosses over to the other side to the thalamus. The right side of this eyeball comes all the way to the chiasm, but then is bounced back into the right side. So all of your left visual field goes to what side of your brain? The right. If you lose the optic nerve here, do you lose your left and right vision? No, because you have left and right vision from this eye. All you lose is the ability to see some of the stuff over here way at the extreme. If you lose the chiasm right down the middle, do you lose the ability to see out of either eye? Can you still see in both eyes? Yes, but what happened? You only see with half of each eyeball. So if we cut the chiasm right down the middle, anything that hits this part of the right side of the left eyeball doesn't make it over. Anything that hits the left side of this eyeball doesn't make it over.
So you basically have peripheral vision.
Exactly. You'd have a big blind spot in the middle. You would just have peripheral vision.
I knew a guy in school that was like that.
Did he read, like, he'd put the book over here and read, like he's looking at the table and
He
read?
had a machine that he put the book under that, like, way blew it up. And
Oh.
he would still, like, have to turn his head. It was weird.
My grandmother had macular degeneration, and it actually destroyed her central vision. And she would read. She read the Bible every day, and she'd have to. Yeah, she'd be looking at the table, and she'd hold the book in her right hand
Yeah.
to read it. It was unique. You never say crazy when it's your own family, right? Oh, wait a minute. Actually, that's exactly the opposite. I say crazy about my family a lot. But you can see the different parts of this pathway. So here you would lose vision from all that eye. Here you would only lose part of the vision from each of the eyes. If it's destroyed here, what happens to your vision? What do you lose? Both eyeballs are perfectly healthy, but what did you just lose? You can't see anything on the right side of your body because it's not getting through. Both eyeballs are healthy, the pathway is coming to the chiasm, but all the signals that are coming from your right visual field are getting cut off. And then at the thalamus, this is another one, if you cut after the thalamus, remember that blind sight that I was talking about, where people were completely blind? It's because the signal didn't make it to their conscious awareness in what lobe? The occipital lobe. Yeah, what optic lobe? That's the first time I've ever seen that. That's from your book. But, yeah, the occipital lobe
It says
back
that
here.
in our notes, too.
Yeah, you know, maybe it's because I'm a neuro person. I catch a lot of little flaws in your book in the neuro section. But so it comes back to the occipital lobe. With the blind sight, their signal's getting to the thalamus. What's the thalamus? It's your relay station. So it's relaying to all over the brain. But the main conscious signals for vision aren't making to the occipital lobe. So they can instinctively, in their limbic system, kind of weave a little bit to get around things. If you ever get a chance to YouTube it, just YouTube blind-sightedness. And you should see a video on it. It's really cool. Right. Another interesting thing is that some people are colorblind because their eye doesn't have the receptors. Some people are actually colorblind because they have damage to the color receiving area in their optic lobe. Now I'm doing it. In the optic region, their occipital lobe, in the optic region. So there's so many things that can go wrong. That's what's kind of cool about pathophysiology is that you learn the pathway and there's so many things you can break. It's really hard in medicine to say, I have this. Oh, you definitely are. This is what's wrong with you. You have to like slowly eliminate all the other possibilities before you can actually diagnose with something. So it's really fascinating. That's why I love that show House. I love that show.
So if boys that are, like how a lot of boys are born with color blindness, what?
That's usually the eye itself. That's the retina. Because there are about 20 types of color blindness that affect the eyeball. And that's actually, when you say it's mostly boys, that's an X-linked trait, which if you go into pathophysiology, or if you guys want it, we already talked about that in pathophysiology, but it's an X-linked trait, which means girls can get it, but boys get it more often because boys only have one X, where girls have two. So they get the bad flaw. If you're a girl, you get one bad gene, you have a backup, you have a spare. You just turn on a good one and forget the bad one. But a girl, if she gets it from mom and from dad, she's colorblind. That's why it's more.
Seems like a lot of guys are colorblind.
Yeah, my son is.
Completely, or just like
Yeah,
red
well,
is.
You said your
colorblind?
son is colorblind?
Yeah, but it's like all the boys in our family have it. Like my
And your family, not your.
not my husband's family.
Right.
So
What's interesting about colorblind is there's absolutely no way that the dad can pass it down to the child. It always comes from the mom's side.
And the mom's never the one that's going to have. That's our
Right, and the
daughter.
mom rarely has it. Yeah, if a mom actually is colorblind, 50% of her kids are gonna have it.
But it's funny because Riley never knew he had it until like we figured out that he had it.
Right.
Yeah.
You know
No,
why?
that's not very.
He figures out the colors on his own, and he'll guess his colors, right? Because he learns what the color looks like to him.
Right.
So, but unless like red and green are together, like he can't distinguish certain colors, like when they're right next to each other or blended, or
But when you're taught
Because
what
his
red,
world.
It doesn't matter like what color
right,
it is.
you used
You're
to
just
see
taught
what
that.
your red
Yeah.
is. Yeah.
It's his world. He's creating his own perception from his
Yeah.
world. He has receptors for a specific thing, and that's all he knows.
Yeah, it's
so
kind of interesting.
it's cool.
I always thought
And it's proof that you create your world. So, thank you for the support there.
You're welcome.
And then we already talked about this. This is just, if you go back to this slide and you look at number 1, 2, and 3, if you knock this out, you can see what happens to their vision. So like we were talking about before, you lose certain areas based on where you cut the connection. But it's a pathway. If you shut any part of that pathway, you get different effects. It's kind of cool. All right, so next we'll talk about color vision. In color vision, you only see 3 colors. And by the way, color is technically a lie. It's not real. When you see colors, you're actually seeing bursts of light. It's just intensities of light, photons of light. When they hit these different cones, they hit different ones differently. When you have a blue cones hit with a certain photon, it's only sensitive to that photon. When the reds hit, it's sensitive to only those specific photons. When you have a green, it's those specific photons. So when you see different spectrums of light, they're all getting activated at different intensities. And I'll show you what I mean by that. So here's what we've always learned. How do you remember these colors? Roy G. Biv. So red, orange, yellow, blue, green, blue, and then indigo, violet. When you see red light, what's happening is that the light wavelength, the photons of light, are closest to this frequency. So that cone is actually being stimulated the most, and you see red. When you see something like orange, right in between yellow and orange, you're seeing a little bit of red light, you're seeing a little bit of this green light. This one's being stimulated just as much as that one. So your brain interprets that this color must be right in between those two. And when I said color is kind of a lie, when you think about this, how many people have played with finger paints? When you mix red and green, what color do you get?
brownish.
Turd. Right? You get this ugly brown color. But if you mix red and green light, which I had time for, I have these color slides and I was going to put them up on this, but if you take a red and green slide and you put it behind a strong light source, it actually turns yellow on the wall. It's cool because light actually behaves a little bit differently when you put these slides in front of it. So if I were to show you a red and green light, you'd actually see this yellowy borderline orange. If you see blue light, a strong blue, it's because this is the only wavelength that's really being stimulated. Look at this. So you can look at the percentages. If you see a blue light, only the blue cones are being stimulated, and your brain knows this is blue. It's just a label we give to that intensity of light. If you're seeing green, you're seeing primarily this, but it's a little overlap of that, a little bit of overlap of that, but primarily right here down the center. If you're seeing yellow, you can see the red and the greens are evenly split. This much and this much are equal. So your brain says it must be somewhere in here. So this is always fun. We can actually burn out those receptors. They change or adapt quickly, so they're called what kind of receptor? Tonic or phasic? Phasic changes really quick. So if you focus, I'm going to turn the lights off, but I want you to focus right at this. So you just keep staring.
Are you gonna scare the shit out of us again?
No, I'm not going to scare you, I promise. I want you to get involved. So you focus on the nose. Keep staring. And keep staring at it, and then I'll pop up another background. Do you see the yellow happy face?
Blue.
So what happened, I'll go back to this, I'm stimulating these receptors. They're phasic, right? So as you're staring at it, they're like, oh, we know it's still blue, and they shut off. They actually just completely shut off. So when you put a white screen in the background, I'm stimulating all of these. White light stimulates all of these different frequencies. So now the blues are tired, they're burnt out. Which ones are you stimulating? You're only stimulating the green and the red evenly. So what color should you see?
I was
Yep.
saying blue.
If it's half
blue
this and half
like,
this.
Like the blue was the eyes and the mouth, and then there was nothing there.
Okay, the eyes and the mouth might be a different color. What I was trying to do with this one is I put the white here because what's the opposite of white? Black. And this is as white as I can get it, so I just tried to get it black. But you had a little bit of overlap with this. How about this?
Blue on blue.
And just stare at it for like 15 seconds. Now what is it?
Yellow.
So it's a little bit more brilliant yellow. The background actually changes the way that you perceive it just a little bit. So what should you see here?
Yellow, red.
So right now there are only two cones that are being stimulated. What are those two cones?
Yellow and black.
Yellow.
Red
Red and green, right.
What?
So it's the red and greens. You only have red, green, and blue. So you're stimulating the red and green right now. You're burning them out and you're making them tired if you're staring at the nose. So when I give you a white background, what's the only cone that should be able to be stimulated? Blue. So you see like a bluish color face with a sad white mouth? What's funny is that this is the same color as that. It looks red though, doesn't it? It's crazy. Oops, there we go. And then people have manipulated this. When we learn about how these things work, if you focus on the arrow again, we'll do it for like 15 seconds. They've actually taken different tones of these different colors so they can get the perfect blend. So they make the colors just right when you see the after image. So if you focus on the middle of that, we'll do it for another 15 seconds. And you should see like the perfect blue. You should see the perfect red. And of course where the black was, now it's white. And I put some of these images on here, like this little light bulb. If you stare at that, later you can do it. If you stare at that and you have a white piece of paper underneath, wait for like 30 seconds and then pull it out. What's going to happen to the light bulb?
It's going to be opposite.
It's going to turn on, right? It's going to be white instead of black. And this is kind of a fun one too. So we can make the image even more perfect by, if you stare at this, same thing. We'll do it for another 15 seconds. Look at that little white dot right in the middle. But I'm going to tell you I'm going to flip it. Don't blink until I've actually flipped it. So I'm going to flip it. You keep holding it. You'll see colors. And anytime you want, after you've seen the residual colors, then you can blink and see what happens. But just keep staring at that circle. And I'm going to change it in 5 seconds, but don't blink. If you have to blink, do it now before I change it. 4, 3, 2, 1. Don't blink anymore. And then anytime you want now, you can blink. What happens as soon as you blink? Yeah, so the color stays until you close your eyes and everything resets, and then you open it again, and then poof, it's black and white. Yeah. And I put some of these you can do on your own, but I love this as a little beach scene. You can see the perfect blue skies and kind of teal waters and everything. It's awesome. Okay, so the visual information is just perceived by your eyeballs, and then your brain has to actually interpret it. I'm the great and powerful Lord! So, I watched that movie the other day, and now it's in my head. But when I walked back there, that's how I felt. I should put like the smoke and everything, the picture of my face and the smoke. Okay, so anyway, the visual image is actually modified. It's a hierarchy. You get some processing in your eyeball because it takes it from this 120 million basically pixel image, condenses it down to 1 million pixels right in the eyeball. So before the image even leaves the eyeball, it's modified and refined. Then it goes to the thalamus and it's weeded out. What do you not need to see from this picture? And you can focus on one thing in the middle. And then that image is sent back to the it's sent back to the occipital lobe, the optical cortex, all the way at the back. And then it's processed one more time. You actually see the image. And the image that's back there, if you get a chance to watch the videos, there's, I think it's the perception one, that actually showed you how an image is split apart. When you see Grandma's face, you don't see Grandma's face. You see line, line, lines, 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 and they're all over your brain. You have to put those pieces together to see her. So it's a large, complex process that's happening. Depth perception takes the fact that one eye is slightly off center from the other, and your brain interprets that this gives a little bit of a dimension. So when you're looking at something, it gives more of a three-dimensional texture to it. That's why they say don't drive when you have to cover one eye, because you're not used to the three-dimensional environment. You think, oh, I can do just fine. But in reality, if we do a test and I have you cover one eye and I start throwing eyeballs at you, if I start throwing baseballs at you, you'd have a way tougher time catching the baseballs because your three-dimensional perception's off. Okay, so like you see, one eyeball is shifted off just slightly from the other so that your brain can interpret depth. There's a reason that our eyeballs are at the front too. Do you know why? So if you look at something like a sheep, their eyeballs are more to the side. Or a goat, their eyeballs are to the side. Why are tigers and lions and humans, why are our eyeballs at the front? We're predators. We have great three-dimensional imaging. But the animals that are the prey have it more to the side. Why?
So they can see what's coming up.
They see something sneaking up behind them a lot better. Yeah. So I love these things. How many people cannot see these? It ticked me off. Jay and Silent Bob, when they did Mall Rats. It's a schooner. That thing, that's how I felt. I was so pissed when people could see it and I couldn't see it. And then I found this book that showed you how. And the trick is that you take this block, and you kind of blur your image until a third block actually forms in the middle. So you see all 3 blocks. You guys know what I mean? You kind of like relax your eye and kind of cross it a little bit, and then poof, there are 3 blocks. You slowly drift your eye down, and as you're drifting your eye down, you relax a little bit more, and the image just starts kind of popping out at you. It's been a while since I actually looked at that.
I think
and
it's
then,
hard from far away.
I can't see these. I've never used them.
Oh, I see it.
Well, that's because he's doing
the first time I did that, I totally didn't mean for that to happen, but somebody did that. They're like, oh! I was like, what? Oh, I get it. But now it's just kind of fun listening to see if somebody does do that. But yeah, that's what happens is the image will start popping out, but of course they'll have all this weird background surrounding it. But it just slowly pops out at you like that. And it's basically that you're making your eyes lazy and they're shifting off their normal 3 dimension, and then this image pops out at you.
How come some people can't see it? Like, I can't.
You just haven't learned how to see it, that's it. It's a process. You're stubborn. Are you a stubborn person?
Yeah.
There you go.
I just can't, like, I don't know. I can't even see the block in the middle.
Go online and look, like, Google how to do it. So they'll teach you a little bit more about that. And once you see one, though, by the way, so that works sometimes for people too. I stare at it and I relax as if I'm looking at the back wall, even though it's right here. So I relax my eyes as if I'm looking through the picture, and then somewhere in that shifting, it starts popping out at me.
Say, like, those are the same things as 3D books that you can.
Magic Eye.
Put it really close to your face and then move it back.
Yeah, that's the same idea. So you can either unfocus your eyes or you just move the object. But it's. Yeah, you have to let go of control. You have to let go of the control. What's funny, though, is how many people, once you saw it, you usually don't have a problem seeing another one. It's once you get it, it's there. It's the same as physiology. Sometimes you're like, oh my God, why can I not get this? But it's funny because once it clicks, you go, oh my God, why could I not get that before? And then everything kind of clicks from that point on. So this is a little bit harder. This is actually 3 rings. So if you look at it on a smaller screen, usually it's a little bit easier to see. So that's vision. Let's go on to hearing. So hearing, here are the functional things. Remember the structures for hearing. What do you call those little receptor cells for hearing? Do you remember from anatomy? Nobody remembers, huh? Call them hair cells. So what's the name of the structure that has the hair cells in it for hearing? Looks like a snail. Cochlea. Yeah. And then when we look at the pathway, actually I'll talk about each step, the auditory tube and all of that. But what you're hearing is actually 3 textures of sound. One's called the pitch. And the pitch is the tone. So if I go to a keyboard and I go bing, 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 and I keep hitting the C key, I'm hearing the pitch. If I go, is it 12 keys higher? Yeah, it's 12 keys higher on a keyboard and you hit the same key, it goes bing, bing. Well, I can't do it now. It's a higher, it's the same pitch, but it's just a little bit higher. Right? So it's still a C, it's just a higher one. Intensity is if I take that same key and I go bing, 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 and I go bing, 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 bing. Did it change its pitch? No, it just got louder, right? That's the intensity. And timbre is the quality of the sound. If I take a piano and I hit the C on the piano, and I go to a guitar and I pluck a C on a guitar, do they sound the same, or can you distinguish a piano from a guitar? Yeah. This is the timbre. There's a quality to the sound that's different. Even a drum has a C on it. I mean, whether you can imagine it or not. You can play a C on a piano, you can play it on a tuba, you can play it on a guitar, you can play it on a drum. This is the timbre that makes it different in quality. So those are the characteristics, and your brain interprets these different characteristics by blending what's happening at those little hair cells. So here we go. Here's how the air travels. So the sound travels through air molecules. It's bumping them like billiard balls. Ding, 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 in a long chain until they finally go down your ear tube, your auditory canal. So as it's bouncing along the auditory canal, this is designed like a funnel to try and bring that sound in and focus it to a point. Right? Now remember, your ear's pointed this way for what reason? So you can hear it in front of you better. It's the same reason we have eyes in front. We're predators. We want to know. We want to be able to focus. If I make a loud sound, where do your eyes go? To wherever that sound was. Is it easier for you to focus if it's somewhere in front of you or somewhere behind you?
In front.
in front. If you've ever done that, like something's making noise behind you, you like turn around and you're like, okay, it's somewhere here, right? And then you have to slowly narrow it down. But if it's in front of you, automatically your eyes start going right to that region wherever it's at. Unless it's a cricket. Yeah, I know. Those things are so stinking. Urgh. Thanks for dragging those fingers on a chalkboard for me. But so the sound's bouncing along in here, and then that sound vibrates this little, it's like a cellophane membrane. It vibrates like this. As it's shaking it, the back of that membrane is connected to 3 little bones, the incus, the malleus, and the stapes. So remember the mallet, the hammer, and the stirrup. And they describe the shape like that. It looks like a stirrup on a horse's saddle. And then the mallet smacks on the anvil, which shakes the stirrup. So it vibrates these. The whole purpose of these 3 little bones is amplification. So they amplify the sound. They take that signal from a weak signal and they amplify it. They turn it up. And then it vibrates this little thing called the oval window, and then the oval window will actually vibrate fluid inside this coiled up snail structure. It's coiled up as a long tube. So it starts and it keeps going all the way back until the very tip. It's one long tube that's just coiled up. As I vibrate water here, stronger pitch or stronger intensities will actually vibrate further along here. And I'll show you how that works. But it's a 3-step process. The outer or external ear vibrates the tympanic membrane, which shakes the middle ear ossicles. For what purpose? Amplification. What's in this, by the way? Water, air?
Air.
It's air. If you get water in here, what happens to the vibration of things underwater? They're muffled, right? If you're trying to listen underwater, everything's it's muffled. When you have a cold, and this is filled with snot, right? I thought it was mucus, but it's snot. Anyway, so when it's filled with snot and it blocks it up, you can't equilibrate that air environment with the outside part, so it puffs up and it pulls on those bones. What's going to happen to your amplification? It's not going to work very well because they can't vibrate appropriately when you pull too tight on them. So everything sounds muffled and you're like, you're trying to hear better. And then, of course, that little oval window moves the liquid. So here are all the anatomy parts. I could care less if you memorize those. You just need to memorize these special structures. The tympanic membrane. It receives information. It takes air waves and vibrates because of these air sound waves.
What about an airplane?
What about an airplane?
Popping your ears.
Is it just the pressure?
It's the pressure. Yep. So when you change, that's what you're talking about, right? The popping the ears.
Yeah,
When you go up in an airplane, if the pressure changes too fast, the pressure in your mouth and throat doesn't get a chance to equilibrate with the inner ear fast enough. That's why they tell you chew gum, because it's actually squeezing that tube and allowing air to move easily through it, back and forth. Okay, so the tympanic membrane receives. The tympanic membrane it separates the external from the middle ear. The middle ear, you have to know the 3 bones. They're called the ossicles, and they amplify sound. They're surrounded by what? Water or air? Air. Changing the pressure in here can actually change your ability to move those bones, so it changes and makes things muffled. And then the last step here, you have the oval window. And the oval window is connected to the stapes, and it moves fluid. It brushes fluid back and forth in the cochlea. So this coiled up cochlea, it's more coiled than this picture shows, but it pushes the fluid in here. The stronger the fluids move, the further down the cochlea it will shake. Right. The next structure, if you look at the center canal through here, it's still filled with fluid, but it has little tiny structures, and this is a slice, so we're going to look across it. This top one's called the tectoral membrane. It's tectoral plates. It's a little thing that vibrates. When you move the fluid, it vibrates. It's a long plate, so it actually pivots kind of like this. But what sits right above are those little hair cells. And the hair cells sit on something called the organ of Corti, and I always remember it because the hair cells are like little keys on a what?
Piano.
Piano, which is also like an organ, right? So when you push on those keys, it stimulates different pitches. The further along that goes, I hear high pitches when they're closest to the window. I hear low pitches when they're way down here. So if I vibrate here, my brain says, well, that must be a high pitch, and it can kind of pinpoint it. You've got about 16,000 of these little hair cells going along. So right down here, if it's vibrating these highest ones, I hear the highest pitch, real high frequencies, that annoying sound. When I feel like a bomb go off, a mile down the road and it vibrates that low pitch, it's vibrating all the way down here, and I hear the rumble. And the organ, I love this because each of those cells, it's almost like a long 16,000 key piano. Highest pitch goes beep, 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 all the way down to the lowest pitch in order. So which ones get the most stimulation, the low pitch or the high pitch? The high pitch. Whether you hear a low pitch or a high pitch, you're still vibrating these. Your brain's just detecting how far down the keyboard is the lowest key. So if I shake the low pitch one, the high pitch ones shake too, but your brain's going, those aren't as important as that one right there. So it looks at the lowest key there and says, this must be the average pitch that I'm hearing. It's a really cool process, the way your brain weeds out this information. So if the high pitch ones are stimulated the most, what's going to happen with time? They die off. They wear out. The more high-pitched sounds you listen to, the more they die off. There was another experiment, kind of like this Sew the Kitten's Eyes Shut, where they took a kitten, put it in a box, and they put one high-pitched frequency on in that box, and the cat's meowing and annoyed by it. But a week later, the cat didn't hear it anymore. It burned out that hair cell, or those hair cells for that pitch. So then they turn a different pitch on, and the cat's like, ah! And then they turn it back to the original pitch, and the cat's fine. Why? They killed off the hair cells. Yeah, they don't grow back. They're neurons. When they killed them off, they're gone. So as we age, we're vibrating these and we're killing off those cells. Once that sound's gone. Huh?
Like a dog's ear, how they hear really high pitches?
Dogs have a higher frequency range. I don't remember how high it is. I know a bat can go up to like 32,000, where we can hear up to 16,000 because we have 16,000 keys, but a bat has like 32,000.
Sensitive?
They're more. They hear the higher pitch. They're more sensitive because not many things out there are higher pitched. So for a bat, they squeak at a really, really high pitch, and that's a unique sound. So they can hear it at that high pitch. It's almost like code that goes over the airwaves. You can only detect the code if you know what to listen for. That's what a bat's doing, is it's listening to the higher pitch range. Where a dog can hear a higher pitch too, because it needs that range. It's listening to other animals for hunting and stuff. We have such a narrow range because we don't use anything outside of our normal. Yeah, unless you're blind, which is really cool, echolocation. They make a high-pitched tick. They go, blind people can do that, and they can hear it bouncing off walls like a bat does. But what sucks for them, I was just watching a video about it the other day. I think actually one of those videos has it on it. Is a guy's going, unfortunately I'm not a bat. I'm blind and I'm a human. I can hear kind of like that, but a bat's more sensitive to differences because it has that bigger range that it can listen to. And this guy's riding his bicycle. He's completely blind, riding his bicycle down the road going the whole way. And what's cool in that video is that the researcher's kid said, I can do this. And he blindfolded himself and actually taught himself echolocation. He can see, but he went around blindfold doing this and learned how to do the same thing. So we can learn stuff like that. All right, and I already talked about the transmission, the pathway. You just need to know the different steps. It's physiology. It's step by step. Sound waves come in through air. They vibrate mechanically through the bones. The bones amplify and move what? What's this substance?
Water.
It's a water substance. It's a thick water. That vibrates more thick water, which taps down that tectorial membrane, which plays the hair cells. Once the hair cells are stimulated, they all go down to the auditory nerve. I've got too many research experiments. Remember the muskrat thing? But it goes to the auditory nerve. So let's see if you caught the different steps. In order for hearing to occur, which has to happen first? Or sorry, which one has to happen at all?
2.
So let's go backwards. The air in the cochlea pushes on the hair cells. Maybe I shouldn't emphasize things, right? What's wrong with that number 4? There's fluid. There's water. It's that perilymph and endolymph. It's thick water. So there's water in the cochlea that actually does that. That one's wrong. How about number 3? The liquid in the middle ear gets displaced. What's shifting in the middle ear?
Air.
Bones that float in air, right? So we know number 3 is wrong. How about number 2? The hair cells on the tympanic membrane bend. The hair cells are in the cochlea. Where's the tympanic membrane?
On the outside.
That's in the external ear bordering the middle. What's actually touching the hair cells? Is it tympanic membrane? I made that sound because I was hoping it would stick with you when I said tectoral plates. It's the tectoral membrane, right? Everybody knows what I'm talking about, right?
Yes.
Okay.
So
Tectoral plates, magma, liquid, magma. It's hard to, with all this hair, it's hard to envision Dr. Evil, but
do you ever listen to your lectures back?
I try not to. I really try not to because I hate listening to myself. But yeah, I'm sure that they're a hoot. All right, and then so number 1, stapes pushes on the oval window, and we know that one's right. Stapes is the little bone that actually vibrates the oval window. My didn't go through 8 years of evil medical school to be called mister. Alright, so pitch discrimination depends on the region. And I already told you this. What's the higher pitch? Where is it closer to? Is it closer to the stapes or closer to the center of the cochlea? To the stapes. So if you vibrate that high pitch like this, your brain interprets it as being high pitch. If I vibrate it way down low, it takes the lower section and says, well, it must be a low pitch because it's vibrating these down here. So remember, frequency is telling you it's the pitch. The frequency is the speed of the wavelength. The faster the speed, the higher the pitch. So let's see how good your hearing is. I hope this is turned up. Oh, crap. How many people have heard this? You do what? Oh, you sneaky kids. Just this last year, I've changed on this. It made me so angry because I used to be in like a lower range than my age, and now I'm starting to age quickly, I guess. I know. Oh, come on. So much easier when a quick link. So I'm going to turn off the projector for just a second. And I'm going to see how many people we hear. So everybody should be able to hear that. Anybody not hear that? Okay, just checking. So that's hitting the mid-range. That's actually at that 8,000. So it's hitting that little 8,000 key. Now I'm going to hit some other ones. And just by a show of hands, you let me know when you hear something. Well, still no, huh? You know, a bunch of deaf people, huh? Dang. Got something finally. Now I know you're fooling with me. Don't forget, I'm a scientist, so I know what a placebo is. You shouldn't hear anything here.
Are we supposed to keep it up? That's what.
Well, if you've already heard it, can you hear something now? Anybody hear anything? That was actually a little bit lower. By the way, if you're 24 and younger, you should be hearing these, according to this test. Everybody heard the 8, though. That was really loud, so I know it's turned up loud enough.
Can you hear that?
No, I can't.
No.
I just know that I'm pushing it because the button lights up when I push it.
Nope. Yeah, I can hear that.
Yeah, if you're under 30, you should hear that.
Yeah, I can.
Thank God, I can actually hear that one. That was awesome. Woo! If you're under 40, you should hear that one.
Yeah.
Under 50?
You can't hear
Shut
that,
up.
Maria?
What are we talking about?
Are you serious?
I can't hear anything.
How many people can hear that?
Oh my god.
Woo!
I've shot a lot of guns in my life.
You can't hear that at all?
Something's going on right now.
If I say...
No, not
Yeah.
now.
Thanks for that little piece of information, not to pick on you. I shot a lot of guns in my life.
I have too, but I guess more earplugs.
Yeah. Don't piss me off.
You can't hear that right now.
So, yeah, you should... Oops, I thought that came back on.
Just gotta be really quiet so she can't hear you.
Yeah.
Wow.
Just
Now
right
I
now.
Okay, well, that's... Yeah, that's...
Back at 8. Yeah,
You wear earplugs when you shoot?
sometimes.
No.
I always
Not when you're killing things in the
Not when
field,
I'm
though, right?
hiking.
Yeah.
Okay.
So, yeah, and everybody should hear that. But,
Can you hear that?
how about that one? Yep, yeah, that one's... Do the 20?
Yeah.
Down here.
No.
Yeah. And they even made a commercial for, I think it was the movie White Noise, where it was all this creepy, high-pitched talking. So kids will listen to it and go, what? And they'll actually listen to the commercial, but adults will sit there and it's just static on the screen. I thought that was creepy. Because, right, because they're the wrong, they're a different pitch. You've blown out that, yeah, so basically the higher pitch, you've just crapped all over those.
Is it like my brother leaves his TV on all the time, and like he won't have the sound on, but when I walk in the house, I can.
Yeah, and thank God I can still hear that too. Like when you walk in, you can hear somebody left the TV on, even though the sound's off, because the TV emits this higher pitch frequency. Yeah. So that's just a test of your hearing. And once you burn those out, they're gone. There's no growing those back. Yeah. All right, so we'll move on from that. So auditory cortex, remember what lobe is that in? Well, here's your ear, so where's it probably at? Temporal lobe, yep. And it's the pathway. It goes from the cochlea down to the audiovestibular nerve, which is number 8. It goes all the way into both sides, actually. So it goes on both sides. But the side that's stimulated first helps you orient to the sound. So if it stimulates this side first, then your brain knows that the sound must be stronger here. What if it stimulates both sides equally? Where does your brain think it is?
In front of you?
Right in front of you, or directly behind you, right? So it's the pathway. You know the pathway. The cranial nerve goes to the brain. And what part of the brain is it going to go to before it goes to the cortex?
Thalamus.
The thalamus. What's interesting about this is it goes to the brain stem. I told you there was a part of the brain stem that was sensitive to auditory and visual. Was it the medulla, the pons, or the midbrain? It's the midbrain. It goes to the brain stem to the midbrain to help you with a reflex to orient yourself before you're consciously aware of what the noise is so you can respond to it. I remember when I was a kid, we were playing baseball and we were gathered around the backstop while it was raining and lightning hit the backstop. Anybody ever had something like that happen? All I remember is that sound, and I remember like opening my eyes and I was laying flat on the ground with my head turned towards the backstop and then crawling as fast as I could away from it. It's so that before I even knew consciously what happened, I was protecting myself and getting away from it. Yeah, it's awesome crazy. And then the cortex is arranged just like the cochlea is, in order from low to high pitch. So it goes in order. If you were to, so I always wanted to take like an electrode, you know how they poke the brain, you can feel things with your finger? Take it and just go across somebody's auditory cortex and see what they say. I wonder if it sounds like a piano when somebody strings their finger across all the keys. Right. And then the other device, and there's vestibular apparatus. And the vestibular apparatus is there to help you with balance and coordination. You've got 3 semicircular canals in here, and they are oriented on 3 planes. One's straight ahead, one's flat, well, off center flat, and the other one's at 90 degrees to those 2. They sit 90 degrees to each other. So any 2 of them, you can isolate them, and they sit 90 degrees to each other. What's cool is that bottom one sits actually 30 degrees off center. And they keep thinking, why is that? But you know what? We're a predator. We run. And when we run, we have to have very precise sense of direction where we're at. So when we run, what do you do with your head? You actually tilt it about 30 degrees. So they think that the reason it's like that is so that when you get down and you start running like this, it actually gives you better balance and coordination when you're moving quickly. It's kind of a cool device. Our brains are incredible. All right, this is how it works. So this is all fluid. It's a thick, watery fluid just like you have in the cochlea. When that fluid shifts, it hits this little thing called the ampulla. And the ampulla has its own version of little hair cells, but they're called stereocilia. So they're in there all vibrating. When they get hit with water, they go boom, and they shift to the side, and then they come back. When you shift your head to an angle, it shifts the water, and it pushes that cell, and it helps you orient that your head must be going this way. All right? So if I hit all the cells over here, on the right side of my head, it's pushing these. Guess what it's doing to the ones on this side? Letting go of them. It's relaxing, just the opposite, right? And so it's shifting and making me feel like I'm falling this way or I'm moving this way. If I shift back, it does exactly the opposite. If I spin around really quick, like that, over and over and over again, and I stop, guess what happens to that fluid in my ears? It's like taking a bucket and spinning around in circles really quick with the bucket and then stopping. The water will stay flat as long as you're spinning, but as soon as you stop, what's it do? It splashes back and forth. So in your brain, it's actually splashing back and forth. It's hitting the right side and then the left side, and then the right and then the left. So the right side's being activated while the left's turned off. And then suddenly the left's turned on and the right's turned off. And it's making you feel like the room's going back and forth. Right. And then the next ones, these are called otolith organs, not ossicles. But they're the round structures here. They're surrounded in those little hair cells all the way around. It's almost like Epcot Center with this big dome. But on the inside, you have hair cells completely covering the inside, stereocilia, technically, covering the inside, so that when you shift your head, you can have a full 360 degrees, forward, backwards, whatever, sense of where you are in space. These are more sensitive to acceleration. So when you accelerate forward quickly, it shifts a little tiny thing. It's basically a calcium carbonate crystal, like a piece of bone in there. And when you accelerate quickly, that bone shifts, and it lets you know you're accelerating. When you go straight up and down, when you drop quickly, that bone kind of hesitates and floats slightly, like if you're dropping in a plane and you kind of lose gravity. And it pulls off, and it tells you where you're at in space. So the two are called the utricle and the saccule. I always remember the saccule is the one for up and down acceleration, like in an elevator. When you get an elevator, push the button, and it feels like they fall out from underneath you. That's the saccule being stimulated. That little crystal floats for a brief second, and you're like, whoa, where am I? You lose gravity briefly. Saccule. The utricle is for forward and backwards, like when you hit the accelerator on your car really hard, and if you have an 8-cylinder, or if you're driving a Viper with 12 cylinders, that acceleration, the G-force coming back, that's your utricle being thrusted backwards. Like this little kid in his jet fighter. Right? So these are for balance and coordination. And I already kind of explained this. So you can see the shift back and forth, the stereocilia shifting back and forth. That tells you which way the fluid's moving. And you can see with a dancer and stuff like that, when they're spinning around, the shapes of those hair cells start bending all over the place, letting them know where they're spinning to. The response time for them gets better, not because the vestibular apparatus changes, but what structure in their brain gets used to those changes and rapid changes for balance and coordination. Cerebellum. Their cerebellum is more sensitive. It has more connections to the neurons, so it's more sensitive to changes. But when the fluids are shifting like this, you can see it bends it to one side, and they can feel which way they're turning a lot easier. When they come to an abrupt stop, they have better control because their cerebellum is more responsive. So if I asked you the portion of the vestibular apparatus that's responsible for vertical balance, not acceleration. Remember, you want better balance and control when you're doing what? Running really quick. What are the devices I told you were associated with keeping you coordinated? Semicirculars. Remember, acceleration has to do with the utricle and the saccule. And I don't need to do a summary because we just walked through all of it. This is kind of cool. So let's trick your eyes and your mind. What'd she say? Look at her again. What'd she say? Same video. That just... Now what's she saying? The whole time she was saying ba ba ba. So look at her mouth. She's actually saying ga ga ga. If you were to plug your ears, it looks like ga ga ga. But they dub over it with ba ba ba, and you hear da da da. Because your brain is trying to figure out, whoa, the visual signal saying one thing, the auditory signal saying another, so it kind of blends it together and says, well, she must be saying this thing in between. So it doesn't matter how many times you look at it over and over again, your brain will tell you exactly the same thing. These signals are overlapping, so it says it must be somewhere in here between. It just takes the average. And it's just more proof that your assumptions actually become fact. You could swear. People would swear they're hearing that da, 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 but in reality they're hearing ba the whole time. Okay, and then taste. Now we're going to do those taste tests, and we're kind of running low on time. But the taste receptors are called taste buds. Yeah, so down in the buds themselves, you have these little specific receptors. So you have the pore at the opening, and you have these receptors. They're connected to neurons. The receptors are specific to chemical signals, not movement, not temperature. They're sensitive to chemicals. Right, so the chemical actually has to go in, bind to the receptor, and turn on the receptor for you to sense it. And we used to think, well, the receptors for sweet are just at the tip, the receptors for sour are just on the sides, and the receptors for bitter are just on the back. This is the old model, but what they find is that the receptors for all those different tastes are evenly divided. They're all over the place. And you have 5 specialized receptors. The salt receptor is sensitive to, guess what? Sodium chloride. So if a sodium chloride molecule gets down into that receptor and binds to the receptor, it sends a signal to your brain saying salt. The more of those salt particles that bind, the saltier the thing tastes to you. If you had a really dry mouth and you took a block of salt and put it on your tongue, you actually wouldn't taste anything. You'd feel the texture touching your tongue, but you wouldn't taste the salt. What do you have to introduce to the salt to be able to taste it? Saliva to break it apart and get that particle down into your taste pore. Sour is sensitive to, anybody know? Acids. Yep, hydrogen ions. The more acids in something, the more sour it tastes, like oranges. So that citric acid, lemons have more citric acid in them. So you bite into a lemon, it activates more of those receptors. So sour turns that on, or acid turns sour on. Sweet sensitive to
Sugar.
yeah, sugar, like glucose, monosaccharides, fructose, glucose. What's also interesting is that lead salts bind to the exact same sweet receptors. Does that explain anything to you? Think about it. Why would lead salts cause problems? Because kids eat paint that has lead in it. What do they think of it? It tastes sweet to them because it's activating the sweet receptor, so then they eat another one and another one. It's like eating candy, but it's actually killing them. So it has a sweet taste, and these lead salts bind to the same receptors. So sometimes there's a mistake in the body. Lead's bad for you, but it just happens to trigger the same sweet receptors. If sour is because of acid, what do you think bitter is because of? Bases. Things are called alkaloids. Things that have alkaline, like caffeine. Caffeine's an alkaloid. Nicotine's an alkaloid. Things that have a bitter, strong bitter taste are full of bases. Baking soda has a really bitter taste to it because it's a base. Alkalines. Dark chocolate is an alkaline. And then umami is that the Western world kept defying that this was here, but the Eastern civilization knew it because they actually put things that are full of a chemical called glutamate that triggers umami in their food. But it has a meaty, savory taste. These meats that you eat are full of glutamate, and when they stimulate those receptors, you have this savory taste. So like aged cheeses or meats. And of course, MSG, monosodium glutamate's a seasoning they put in food to give it that savory or meaty taste. What's interesting is all these receptors will actually send signals up to your amygdala that triggers like instinctive behaviors. And the same thing with your nucleus accumbens, it's addictive behaviors like sweet and sour. Or sweet and sour. Sweet and salty, thank you. So what we're going to do is we're going to actually mix a couple things. I want you to take a shot at the purple and tell me what you think it is. So take the purple. Actually, I don't want you to say it out loud. I want you to write it on the paper. Pick up the purple, drink it real quick, taste it, and then write down what flavor you think it is.
Can I have a piece?
Oh, yeah.
I don't know, it just tastes like sugar.
Which is ironic because they're actually sugar. Okay, and then take the blue one. Taste the blue one and then write down what you think it is. Don't share what you think it is, just write it down real quick. And then take the green one and do the same thing. And then you get the pattern. So take a shot of whatever it is, write down what you think it is, go on to the next one. Take a shot of it, write down what you think it is, and move on. This is actually a test done with chefs that are supposed to have a really keen sense of taste. And what they found is that most chefs can't distinguish the flavors if you take a clear liquid, no matter what it is, that has a specific flavor to it. You put the wrong color in it, and it screws with their brain. So, how many people are still tasting? I'll watch. What did you think the purple was? Nobody wants to answer because you're afraid you're going to be wrong.
Strawberry?
So I hear strawberry. What else could the grape or purple be?
Water.
Grape could be pure water. I could be really screwing with you. It tastes like blah. Acid's not an option. This was actually grape. What did the green taste like?
Soap.
Soap.
It had like a
So I hear a citrusy flavor. What did you think?
there's like a mint taste to it for me.
So Jazz thinks it's mint. So far I haven't heard the right answer.
I think it's
So a lot of people think it's lime usually. It's not. It's cherry.
That's why I tasted like cherry syrup.
How about the blue?
Dish detergent.
You're stuck on the soap. I don't know how many bad words you said as a kid that everything tastes like soap. This was, and a lot of people think raspberry. Why?
It's blue.
Because it's blue. We think blue raspberry, which in reality, raspberries are not even blue. But we think of it because people give blue raspberry for flavoring. This one's strawberry. How about the one that was supposed to look red?
Lemon
It actually
lime.
faded.
I thought it tasted like lemon lime.
But it tastes like 7-Up.
Peach.
Oh, come on.
And how about the orange one?
Oh, that's what I meant was lemon lime. Orange.
Orange. And the orange was actually lime.
Yeah.
Yeah.
That's the only one I got.
So this one usually people can pick out why.
Because it's stronger.
Because it's the citrus and the lime that tastes a lot like an orange. But some people have a problem distinguishing between the orange and the lime. But there are a lot of factors that go into what you taste. One's odor, smelling. Another one's temperature, which if we'd done this a little bit earlier, it may have been a little bit better. But texture is another one. And how many people here can't stand sushi because of the texture, or bananas because of the texture? So, yeah, the sushi and the bananas throw a lot of people off. They just don't like the texture. So they're like, oh, this is gross, even though it's delicious, right?
That's
and then
like those goo packets.
psychological goo packet.
The goo. You've never had goo packets?
I've never had goo.
Do you work out? Oh.
I
Well,
look like
well, you're not fat. I figured you probably.
Anyway, the psychological factors, like pregnancy does this to a lot of women. When they're pregnant and they eat something and they get sick because of morning sickness, not because of the food, then they don't want to eat that food ever again, right? Or if you've had a really bad association with a food, you never want to eat it again. But the last one here, question. So which in the following taste receptors do we not have? Musk, right? We actually have a smell for musk, but not a taste. And we already did this. But most people associate these because this is what we've been learned or educated that these associate with colors. Which blue just baffles me. Evolution didn't tell us that's blue. And then smell. Sorry, we'll do that jelly bean one and we'll be out of here. With smell, it's exactly the same as taste with the pathway. You have two routes. You have subcortical and you have thalamic. Smell is one of your most powerful instinctive drivers of the limbic system because it goes straight to the limbic system. Amygdala, the nucleus accumbens, the hippocampus, goes right through to the limbic system and triggers instinctive feelings. You ever walk into a room and you smell something and you're just like, ugh. Before you even know what it is, you're just either like excited about it or totally repulsed by it. And in fact, sometimes you walk into a room and you just walk right back out because the smell does that to you. And you don't know why. Or you walk into a room and you smell pumpkin pie and suddenly you have memories of grandma's house at holidays like Thanksgiving and Christmas. It went to the limbic system. Why does it trigger a memory? Because the limbic system is associated with memory, emotions, and motivation. That's a subcortical. It bypasses the thalamus and goes straight to the limbic system. The other one's the thalamocortical route, which goes to the thalamus and goes up to the cortex, so you can say, oh yeah, that's pumpkin pie. I smell that. I can smell the nutmeg in it, and I can smell the whatever flavors, clove and ginger in it. But the pathway, you've got a little patch on the top of your nose. It's about the size of a postage stamp. These receptors turn over about every 10 days. Roughly every 10 days, you get brand new receptors. It's kind of the cool thing about the brain. So if you know like a pig farmer, and they leave for work every day, or they do the pig farm, and they go away for the day, they come back, they don't smell that their farm stinks, right? But if you take them away for a vacation for 2 weeks and they come back, they're like, ugh, yuck. They recognize it because they turned over all those receptors over those 2 weeks, and then they can smell it again. This is the sucky thing. These are chemical receptors. What has to actually happen in your nose to smell?
Chemicals.
That chemical has to bind in your nose. So when you walk into a public restroom and you smell that smell, it's because those particles have actually gone into your nose, bound to the mucus, and triggered the receptor. But don't worry, because you've got that mucus, so what's going to happen is one of two things. It's going to unbind and it's going to move down your throat and you're going to digest it. Or it's going to unbind and it's going to form a booger up here and you're going to pick it. So either way, it unbinds, right? You'll get rid of it one way or another. But the chemical actually gets into your nose, into that mucosal layer, and binds to it. What's incredible is that most of your taste is driven by smell. So here's what you're going to do. You're going to do this experiment real quick. I give you Mike and Ike's because they're better than jelly beans. But you're going to plug your nose. Oops, wrong plug. You're going to plug your nose. You're going to close your eyes, grab one of the jelly beads, put it in your mouth, and kind of chew it. Don't swallow it. Just chew it and mix it around. But do not smell it. Just chew it.
I already know what color I am.
And then suddenly, breathe. Do you taste it? So when you first plug your nose, what did you taste?
Just like sour.
Should have tasted either sour or sweet, depending on the flavor. It should be sweet because it's all sugar, right? But if it's a citrusy flavor, then they put acids in it to make it taste sour. And then when you go and you breathe, you've got 10,000 receptors to detect the concentration of, was it lemon? Was it orange? Was it grapefruit? Which are all citrus fruits, right? It helps you distinguish. That's why when you're sick, how do things taste? Sweet, sour, bitter, umami, right? They taste blunt. They taste muted, if you want to use that word. It's because you have so much mucus, you can't bind the flavors to your nose. So it's just an incredible process where both have to combine. So when you chew that up, you taste the sweet or the sour or the bitter or the umami, but when you breathe, the air, the aroma comes up and actually activates sensors in your nose. And I swear, thanks for saying the extra couple minutes. There's one thing I want to point out here. Oops, did I go by it? Pheromones. I told you about this at the very beginning. Pheromones are chemicals I think we're not receptive to anymore. They're more primitive, and that's why they say that. We've evolved. We're better than that. But they're sex chemicals that are exposed in the air. When you're ready to mate, you release these pheromones, and a mate comes along. What's interesting is that this mate, if they have genetics that are similar to you, they're actually repulsed by your pheromones. Why would that be?
Because you don't want to be inbred.
You don't want to be an inbred, right? You don't want to be too much like. You want this genetic variability. So they've done these tests, and one of those videos, actually, if you watch it for extra credit, has this test on it. But this guy he smelled different women when they had exercised really hard. They put their smells, their sweaty shirts in jars, and he rated the attractiveness. And then they did genetic tests, and they found that the ones he rated the most attractive were the most genetically different from him. The ones he rated as least attractive were the most genetically similar. They did this with other people too, like times of the month. Women, when they were ovulating, when they were right at ovulation, they had them exercise and put their sweaty, stinky shirt in a jar. And then women that weren't ovulating, they had them do the same thing. And they gave these jars to men and said, which woman's more attractive? Without seeing the woman, and which one did they pick?
The one that was ovulating.
The one that was ovulating. Yeah. And then they did the same thing with women. The men that had higher testosterone levels were rated as more attractive. Yeah, they made them smell sweaty, stinky underarms. But so we're going to stop there. And as far as the test won't have the last slides, so don't even worry about it. Where it says efferent beyond, none of these questions will be on the test. So I love this picture. You can look at it in the slides. Which of one of these is the real Mona Lisa? The left or the right? This is kind of cool. It was the one on the left. The one on the right, they flipped her eyes and her mouth upside down. And you really can't tell until you look at it straight up. The big picture is of everything is what's your most powerful sense to you? Which one? Which sense is the most powerful? Sight. Sight will tell all the other senses they're wrong, even though they're not. The best sense is actually smell, but sight overpowers everything. Sound, taste, everything.